Hello everyone, so welcome to this second session of the Masterclass listening. So I'm Toulouse and online from Montpellier and Perpignan. And the first talk for this session is given by Sébastien Déjean. And he's going to talk about data science in interdisciplinary research projects. Thank you, Sébastien.
Okay, thank you. Thank you. Hello everyone. So I'm truly happy to be with you today. So with students and colleagues from Montpellier, Perpignan, so great. And so I'm going to talk about data science in interdisciplinary research projects. So I am a member of the Mathematics Institute of Toulouse, and with specificity that I am not a professor, I'm not a teacher at the university, I am a research engineer. So first I will describe you what is my job in the mathematics department, and after we'll go through several interdisciplinary research projects on which I've been involved. So if you have any question, either here or and remotely, do not hesitate to interrupt me and ask the question you want. Okay? Good. So first, who am I? So here is a short resume of me since I obtained my A-levels in a few years ago, 1993. So the name changed, but now in France you can have something related, the same kind of the A-level I added following mathematics and physics. After I've come to this university to follow Bachelor of Science lectures based on industrial mathematics. So it was a kind of, I don't know, revelation for me, kind of epiphania maybe you can say, because mathematics was very abstract in my mind at this area, and industry is where you build some stuff, okay? You have company, they build something. So when I've seen these two words, industrial mathematics, I was thinking that, okay, mathematics could be useful to build something. So that's what I've decided of my future career in applied mathematics. So after I obtained master degree, then I followed during a PhD. I was working on, so you see the translation of the title of my PhD, so comparison of estimation methods for nonlinear mixed effect models. And the application was dedicated to using remote sensing images to monitor culture, gross culture. After I've been hired here at this university as a research engineer in statistics, and a few years ago I have obtained my habilitation thesis to be allowed to supervise thesis, PhD thesis. Okay, so my job, research engineer in statistics, officially you can find it on this website. It's the official definition, official description of my job here at the university. So when we are research engineer in statistics, we can be responsible for statistical aspects in a research laboratory. We can manage statistical projects. An illustration is that we can have to define a data collection and management plan and the associated processing chain. We can also participate in national and international research projects and associated publications. So you have other aspects of definition of the job. In practice, I do like to say that my job consists in answering questions in the real world. Okay, so big. Huge purpose, but I'm going to show you to detail, to give you some example of this. So first, in this book, so How Not to Be Wrong, The Hidden Math of Everyday Life by Jordan Ellenberg, is a professor in mathematics and statistics in the US nowadays. So he wrote this kind of stuff. So I blame word problems they give a badly wrong impression of the relation between mathematics and reality. So he gives an examples of marbles, sharing marbles. So Bobby has 300 marbles and gives 30% of them to Jenny. He gives half as many to Jimmy as he gave to Jenny. How many does he have left? And what he said, what he writes, that looks like it's about the real world, but it's just an arithmetic problem in a not very convincing disguise. And after, he explained what is real-world question. So real-world questions are not like word problems. A real-world problem is something like, has the recession and its aftermath been especially bad for women in the workforce? And if so, to what extent is it the result of Obama administration policies? Well, not so easy as sharing marbles. So your calculator doesn't have a button for this. Now you can tell me maybe ChatGPT can have a button for this, but it's not truly math. Because in order to give a sensible answer, you need to know more than just numbers. And after, with a slice of provocation, it's only after you have started to formulate this question that you take out the calculator. But at that point, the real mental work is already finished. Dividing one number by another is mere computation. But figuring out what you should divide by what is mathematics also. Okay? Just another quote by John Tukey, a statistician, 20th century, wrote this very nice paper to read afterwards, so The Future of Data Analysis, written in 1961. And what is written, I do like this quote. So the future of data analysis can involve great process, the overcoming of real difficulties, and the provision of a great service to all fields of science and technology. With it, that remains to us, to our willingness to take up the rocky world of real problems in preference to smooth road of unreal assumptions, arbitrary criteria, and abstract results without real attachments. So, I don't know what you prefer, considering a rocky road of real problems or a smooth road of unreal assumptions, arbitrary criteria, and abstract results without real attachments. I am not trying to make an opposition between applied mathematics and theoretical ones, because if you want to go to the mountains for a nice rocky road, you can be happy to have a highway like that to go to the mountain. So both mathematics are of course relevant. Okay, so here is a real world problem. How is global warming affecting plant growth? We'll go back to this problem in a few slides, but first, let's go back to linear algebra. So once again, I think I have only one more verbose slide after there will be less text. So Searle wrote this book in 1966, Matrix Algebra for the Biological Sciences. Yep. So what is written? The biological sciences are today in the process of changing from being primarily descriptive to being very much quantitative. As a result, biologists find themselves confronted more and more with large amounts of numerical data. But the mere collecting of recording of data achieves nothing. Having been collected, they must be investigated to see what information may be contained concerning the biological problem at hand. And after a second paragraph, frequently, however, biologists have to subject their data to more complex calculations, requiring procedures that involve mathematical details beyond their general experience. In order to carry out the mathematics, the biologist in this situation must either learn the procedures himself, or at least learn something to the language of mathematics that he may communicate satisfactorily with the mathematicians whose aid he enlists. Okay? So let's have a look, a kind of maybe reminder of matrix algebra. Probably you have already heard about eigendecomposition, right? Yeah? So you know that if you have a square matrix, you can decompose this in a V lambda V transpose with eigenvectors and eigenvalues that you have on the diagonal of the lambda matrix, right? Okay. Maybe you have also heard about singular value decomposition. Okay? Singular value decomposition for a rectangular matrix, M times N. Here you have left and right singular vectors corresponding to singular values. And you have this definition, sigma is a singular value of M, if and only if you have two vectors, U and V, so that you can write MV equals sigma U and M transpose U equals sigma V. Okay? Okay. If you know eigendecomposition and singular value decomposition, maybe you have also seen the link between both. If you start with singular value decomposition of M, U sigma V transpose, you can compute M transpose M and M transpose, and you can link the singular value decomposition of M with the eigendecomposition of M transpose. Okay? Not so difficult so far, so you just have to replace M with SVD decomposition using the property of transpose of a matrix product, and after consider that U is a unit vector. Okay? So now we have this, I would say, solid background of linear algebra, and now we are going to see how it can be useful for statistics, for data analysis. So in data analysis, we have this very famous method, principal component analysis. So principal component analysis is an orthogonal linear transformation that projects the data in a new coordinate system such that the greatest variance of the data lies on the first coordinate, the second greatest variance on the second PC, and so on. And the link between this method in statistics and linear algebra is given here because it can be shown that the greatest variance is the first eigenvalue of X transpose X. X is our data matrix. And transforming coordinates is done using the first eigenvector. Okay? So we have this bridge, I would say, between linear algebra and statistics, and in statistics, in data analysis. So the last verbose slide, it's just from Wikipedia, the page for principal component analysis. And everything is wrapped up here. So singular values in sigma are the square roots of the eigenvalues of the matrix, X transpose X. Okay, we have seen this. And the link between linear algebra and statistics is here in the second sentence. Each eigenvalue is proportional to the portion of the variance, more correctly, of the sum of the square distance of the points from their multidimensional mean that is associated with each eigenvectors. So on. So in boldface at the center of this paragraph, so PCA essentially rotates the set of points around their mean in order to align with the principal components. This moves as much of the variance as possible, and so on and so on. Okay? So we have this tool in statistics for data analysis that relies on linear algebra. But when I organize training workshops for biologists, I avoid to use this kind of terms with eigenvalue, singular value decomposition. It will be a little bit difficult to communicate. So I use this kind of stuff. So it's a question for you. Would you use a cubic box to pack a fishing rod? So, what do you think? No. Mainly, the answer I have is no. Okay? So why no? because we think that if we put the fishing rod in the cubic box is roughly empty, okay? And if I want to pack a fishing rod, probably I will use something like a tube, not a cubic box. So what is the link with PCA, principal component analysis? So let's have a look at this cube. And imagine we have 3 physical measurements: shoulder girth, waist girth, and chest girth. Okay? So I have a few people, I have measured these girths, and I have points like that. Okay? So on the bottom left and foreground corner, I have this kind of body shape, and in the other corner, I have this kind of shape. Okay? So, do we need 3 dimensions to represent these standard individuals? Probably not. We don't want to have 3 dimensions because it seems that all the points are not on the same line, but not so far from a line. So do we need 3 dimensions to represent standard individuals? It's the same. Do we need a cubic box to pack a fishing rod? Right? So instead of living in a 3-dimensional space, I could live only on 1-dimensional one. Okay? Using this dotted red arrow coming from this corner on the bottom left to the upper right. And here, if I project my points on this direction, I have nearly the same information as I had in 3 dimensions. But here I've done dimensional reduction. Okay? And the idea is that I don't have anymore my shoulder girth, waist girth, or chest girth. I have a kind of synthetic variable. We can call it beefiness. Okay? With skinny guys like this one on the left and the tough one like Hulk on the right. Okay? So let's have a look in practice at a very small example. So let's imagine now we have 20 individuals with 5 variables. So shoulder girth, once again, chest girth, waist girth, weight, and height. Okay? So here are the data. It's not a big data problem, okay? Only 20 lines, 20 rows, 5 columns. So let's have a look at what PCA and linear algebra can do for us. So first, I can compute a covariance matrix. Okay? Covariance. So covariance, we have the information related to the variance of the diagonal and covariance between each pair of variables in the other locations. And here we have to keep in mind that the value, the sum of the variances, so 351.9, represent the quantity of information contained in the data. Okay? So, after, what I can do, here I have used my favorite statistical software, the R software. So on the left, I have asked for eigen decomposition of the covariance matrix of data body. And on the right, you can see the same results, just depending on the sign, but the same results using PRComp data body, PRComp for principal components. And what we have, so first, on the left, eigenvalues correspond to the variance of the points projected on each principal component. On the right, we have the standard deviation, just the square root of the values I have for eigen decomposition here, but it's the same information. And after, I have either my vectors on the left, eigenvectors, or what is called rotation in principal component analysis. The idea is that my first principal component, represented at the bottom of this slide, PC1, is defined by a linear combination of the initial variables with the coefficient given by the eigenvectors. Okay? And as soon as we have that, we can compute the coordinates of every individuals on the principal component. Applying the loadings, the coefficient of linear combination we have in the center of this slide, to the centered data. So centering data means for each value, I remove the mean of the column. Okay? So applying this, I obtain the coordinates of every individual on the principal component, and I can check that the variance I can get, what is written here, 255.7, is the highest variance I can compute based on the linear combination of the initial variables. Okay? Is that what PCA ensures? We have the highest variance possible. Okay? So the idea of PCA consists in projecting on the first principal component, and we are sure that we have the highest part of the variance we can collect on a few dimensions. So here, frequently, we can build graphical outputs. First one on the top is called a scree plot. If we represent a bar plot corresponding to variance explained by each principal component. Then we can produce an individual plot. So each individual is plotted, and its coordinates are the coordinates on the principal component. After, we can produce a graphical output for the variables, representing the loadings or the coefficient of the linear combinations to interpret our component. We can also go a step further with the variables, building what is called a correlation circle plot. In this plot, every initial variables, waist girth, chest girth, weight, height, shoulder girth, are represented with the correlation between the variable and the principal component. So here you can follow the arrows on the slides to see that shoulder girth, for instance, on the horizontal axis is located at 0.87, and 0.87 equals the correlation between shoulder girth and PC1. And so on to locate the variables on this correlation circle plot. And one last graphical output we can build to interpret the result of a principal component analysis is this kind of stuff. It's called a biplot. Biplot, the idea is to superimpose the graphical output for individuals and for variables. We have to stretch the cloud of points to be able to do this kind of graphical output. But I would say all the information contained in the data set on the left is graphically represented on the biplot on the right. Okay? So if one day you have to run principal component analysis, you will go through this kind of graphical outputs, and you will have some information. PCA act like you just have a snapshot of your data, so then you can interpret the information that is contained in your data. Okay, so principal component analysis, I don't know if you had already heard about this method, but roughly in statistics, it can be seen in, master, first year master, even before. What I'm going to talk about now is generalization or extension of PCA. So what I've shown you is PCA, principal component analysis, can be applied on only one table full of numbers, okay, numerical data. We can extend PCA in another context. We can consider it's a supervised multivariate approach if we consider not only a table of numbers but also a categorical outcome. Categorical outcome can be healthy people and diseased people and so on, control and treated, wild type and knockout for gene expression analysis and so on. So we can also consider multi-block and supervised analysis. Here, when you have collected several tables related to the same individuals. So we can have it in an unsupervised or supervised framework, multi-block, unsupervised or supervised. And another generalization of these methods is multi-group analysis. So multi-group analysis, you take the 4 framework mentioned above, and you consider that individuals are priorly divided in groups. And you can be interested in finding what is the information that is common to every group of individuals. Okay, so multi-block, multi-group analysis, we have all this kind of framework that can be interesting in data analysis. Okay, so related to the methods I can use to work in this context. So I'm just waiting because I can see people writing some information, so I don't want to change the slide, but you can ask me after if you want information. Okay, so we have seen that PCA, principal component analysis, relies on the maximization of a variance, and we build principal component based on linear combination. If we have 2 data sets, we are going to build, I don't know, PLS components, for instance, or other method, but linear components based on the maximization of a covariance. DA is for discriminant analysis, so the supervised framework. In this case, we can use this trick, so convert a categorical variable into dummy matrix, so that PLS-DA becomes a PLS, so with two blocks of numerical data sets. And after, if we have more data sets, we can have the same principle, we can rely on the same principle, just changing the criterion to optimize. Here it can be a weighted sum of covariance or pairwise covariance between each pair of blocks. Okay? So, another point before, we can also benefit from a sparsity. So, considering a kind of Lay-Su penalty, for instance, we are able, modifying the criterion to maximize, to force some coefficients to be zeros. And if a variable has a 0 coefficient, it does not appear anymore in the analysis, in the interpretation. So it can be useful to have this kind of trick, I would say, to select the most important variables in my problem. So let's go back to our question. Okay. So how is global warming affecting plant growth? So here I'm going to tell you a story based on a project, Voronoi project I've been involved a few years ago. And this project starts underground. So colleagues went somewhere to collect plants underground. Okay, it's a plant model, Arabidopsis thaliana. Maybe you have heard about this. But if you see biologists, all biologists know this plant. Arabidopsis thaliana, model plant, very famous. So once they have collected these plants on the ground, somewhere in the Pyrenees, they gather the seeds of these plants and grow them in controlled conditions. So temperature, light, humidity, and so on. and they are being grown at two different temperatures, 15 and 22 degrees. Once they were up, they collect different parts of the plant, stem, leaf, rosette, roots, and so on. And they put each sample, each biological samples, in these big machines, such as DNA sequencing, mass spectrometer, and so on. And this machine can provide a huge number of figures. Just imagine that this plant can have, I cannot remember, thousands of genes. So this machine can give you a numerical information for each gene of the plant. Okay? So at this point, maybe biologists need the help of mathematician or statistician. Okay? So the data that were collected in this project, we have built an R package. So R is the statistical software, a free one I'm using nearly every day. And we have built a data package so that the data are available for the community. So here they are schematically represented here. So for 60 biological samples, we have data related to proteins of the several parts of the plant, transcriptomics. Transcriptomics means RNA composition of what we are, the biological sample. Metabolomics, so composition of 7 of the plant corresponding to 7 sugars. And phenomics correspond to measurements, number of leaves, height of the stem, and so on, like that. And so we had 5 ecotypes, different types of this plant, grown at 2 temperatures, and we focused on 2 organs, stem and rosette. Okay? And roughly the idea of this project was to run a data integration process. So if I... So data integration can be defined as the process of combining data residing in diverse sources to provide users with a comprehensive view of such data. There is no universal approach to data integration, and many techniques are still evolving. And here is a schematic view on this kind of wheel. If you want to have a complete understanding of a biological phenomenon, you need information based on molecular level, either on the cellular level and about at the level of the organism, and maybe about the environment. Okay? And trying to integrate of the sources of information can give you a global understanding of your problem. Okay, so specifically, here is the kind of question the biologist I was working with wanted to answer. Can we determine a multi-omic signature to classify ecotypes on the basis of floral stem data? So here I have to use my translator from biology to mathematics. So multi-omics means I want to consider all the data sets: proteomics, transcriptomics, metabolomics, phenomics. So I am in the context of multi-block analysis. Signature means that I want to select the most relevant variables inside each data set. So I have to use the sparsity method. Classify ecotypes. So means I have to use a supervised method. So discriminant analysis based on the categorical variable ecotype. And of course, floral stem. So I will filter my data with organical stem. I don't want to work on rosette for this question. So to do so, we run this method: multi-block sparse projection to latent structure discriminant analysis. Okay? There is another kind of commercial name named Diablo, just for, I cannot remember, but the same idea is explained here, okay? It's a paper published in 2019, by colleagues, mainly are in Australia. And here is the same criterion I had put in my slide before, where you have to maximize a weighted sum of covariance between all the data sets taken pairwisely. Okay? Okay, but if I show these to biologists, they say me, okay, but do you want to see how I extract the RNA in my plant? No, I don't want. You don't know what to do with it. So biologists doesn't know what to do with this kind of formula. So we have to provide graphical outputs and help biologists to interpret this kind of plots. And here we can find individual plot in the same way as PCA at the top in the center. On the top right corner, we have the correlation circle plot with variables coming from the 4 data sets. Here on the bottom left, we have a heat map representation where we combine both the biological sample on rows and variables from each data set in columns. And after, we can have also a specific network representation here, so a circus plot representation. Okay, and with this kind of results, biologists can extract biological knowledge from the analysis, the statistical analysis. And so this project resulted in several publications. So in a plant science, where you have the title Cell Wall Modification of 2 Arabidopsis thaliana ecotypes, Cole and Shah, in response to suboptimal growth conditions, integrative study. Another Frontiers in Plant Science on the same topic. In cells, integrative study showing the adaptation to suboptimal growth condition, and so on and so on. And also another that I've been more involved in this one because it was published in Briefings in Bioinformatics, and here represent the protocol we used to analyze the huge amount of data we had for this project. Okay? So here is an example of how math can help to study this big problem of how global warming affects plant growth. Okay? Do you have any questions so far? No, that's okay? Remotely, no question? Good. So now I'm going to switch to other aspects I had mentioned in the abstract. So first one, math can help to give you a shining smile. Okay, not like mine maybe. Does wearing dental braces for instance, 18 months really work? Big question. You can talk to teenagers, they are highly interested in this kind of question. So for a shining smile like this one, you must go through this. Okay, the dental arc, a radio, you have this kind of picture of your teeth. And to know if wearing a dental braces for several months is efficient, it can be useful to model the location of the teeth. And it has been shown, it was a PhD thesis of Maxim Rothenberg. He teaches on the dental faculty at the university, and its PhD thesis was relied on the comparison of several models for a dental teeth position. Okay, and he has shown that the better model we can consider was a 4-degree polynomial without odd degrees, and estimating parameters just like that, with robust linear regression. Okay? So, mathematics, not so difficult here. We have only polynomials, so not so difficult to handle. But it has been shown that it can be useful to help in monitoring the location of the teeth during a process based on dental braces. Okay, another project I've been involved for several years. What are the microbiological mechanisms involved in the cheese ripening process? Okay, in France we are highly proud of our cheese, so we want to understand everything that happens at the microbiological level. So here I'm currently involved in a European project involving more than 10 partners all around Europe, universities in Italy, Hungary, Malta, and so on, others in Belgium, and also with the milk company, milk industry. It was Christian Hansen in Denmark, also NISO in Netherlands, I think. So the idea is to better understand the cheese ripening process. So each of these partners have a PhD student, and roughly it's the same idea as for the Wolomics project. So we have several information provided by several big machines. You know, the microbial composition of the cheese, and we can have information related to genomics, transcriptomics, proteomics, and so on. So the PhD student working on this project can be microbiologists, but we have also in Szeged and in Bologna more mathematics-oriented projects. In particular, Mitya Brisic will defend soon his PhD, and he has been working for 3 years about kernel approaches for the integration of biological data from heterogeneous sources. So the idea is that, okay, what is the distance between these 2 points A and B? Okay, it seems to be easy. Question, okay, I go from A to B. But it's not the only way to measure the distance. We can also have this path in blue. It's not the same length. Another way to illustrate this can be considering this map of France. So in gray, we have the map we know, but in red, what is on this one, on the top, is another map of France, but is based on the duration of a trip in train from Paris. And here, you could see that Toulouse is very far from Paris by train. And Marseille, for instance, and Bordeaux are much closer from Paris than Toulouse. Okay, so if I am interested in traveling to Paris by train, or traveling from Paris by train to other cities, maybe I'm more interested in the duration of my trip in minutes or in hours than in the actual distance in kilometers. Okay? So just to go on these aspects, so here, for instance, I have 2 subpopulations represented by either my yellow round circles, my yellow circles, or my red square. If I want to separate these 2 subpopulations linearly, here it will be difficult. But if we think in projecting this point to another space, it can become easy to linearly discriminate my 2 groups. Okay? I have just some artistic view of this kind of principle. So, for instance, a quote assigned to Lao Tzu, not sure, but things don't change the way you see them. That's enough. Okay? So the idea of kernel methods can be seen like that. I have also, you can think about anamorphosis. So here, it's not so clear what it represents, but if you project it on a cylinder, it becomes more obvious what you can see. Here you have the scary beast, but once again projected on a cylinder, it's just a squirrel with a net. Okay. And I've seen more recently this quote from, so in French, L'écume des jours by Boris Vian. And you have, I think it's before the novel, projection of reality in a biased and heated atmosphere onto an irregularly undulating reference plane, resulting in some distortion. Okay, so what Mitya is working on is this kind of approach is just to project using kernel in specific space where what cannot be done linearly on the initial space can be done after projection using kernel in another space. So what he has done was published last year, so improvement of variables interpretability in kernel PCA. So the idea consists in computing, so kernels is a kind of mathematical function I do not detail here, but it computes partial derivative of the kernel with respect to the variable of interest. You see the direction of the maximum variation associated with each variable for each individual, average value over all the individuals, ranking original feature, and display variables as arrows. So it just the process it developed to enhance the interpretability of variables in a kernelized version of principal component analysis. So it's just a way to see what is partial derivative if we have then several parameters, functions. And what is before also, he has developed with an intern, Mohamed Emida, an R package to provide his methodology to the community. So now let's have a look at the third topic I wanted to talk to you. It's about optimized performance in sports. So here, I don't know if you've heard about this book, Moneyball, and the movie is probably more famous than the book, but it's Moneyball. But the book is also famous because Lisa Simpson, when she wants to show that she's very serious, she's working very hard. So she has equations, F equals ma, but she has also Moneyball. And I'm happy to see that she has also stats in her books, okay? And here, I don't know, do you know Brooklyn Nine-Nine, this series? No? I just show you an extract. Up.
Are you familiar with the story of Moneyball? A man uses statistics and logic to win several baseball games. It's my favorite film. Statistical analysis. It's so beautiful. This all.
So this statistical analysis is so beautiful. It's my mojo, I would say. So he's seen this movie with Brad Pitt, so Moneyball, and he finds statistical analysis so beautiful. So using statistics in sport is more and more common, and a lot of students are dealing with, are aiming to work in this area. So it can be useful to recruit new players, to prevent injury, to model collective behavior, identify optimal strategies, and so on. And here you can have this on the CNRS journal, a lot of articles dealing with mathematics and sport. And here I would just like to talk about this that was mentioned in the abstract Voronoi mesh. So Voronoi mesh is something really easy to understand. You have an area and you have nodes. And so you just highlights the frontier between two nodes, and here the area around one node, separated with the frontiers. Everything inside this area is closer than one node to the others. Okay? And this kind of shape, you can find it in the nature. If you have a look at the leaf, if you look closely, you can see on stuff built by the bees and so on. But you have also this kind of information for Voronoi diagram, and you have here a video on YouTube how Manchester City use Voronoi diagram. Okay, so the idea is that if your players are correctly located on the ground, they control the highest part of the field. So the other team, the opponent, cannot score any goal because you control the big part of the field. What I want to show you here, oops. Okay. So here you have on a real game the Voronoi diagram that is updated in real time. Okay, so you have the blue and the white, and you have the Voronoi diagram updated at each move of each player. You can see each time it moves. So very impressive for me. Okay, and just to finish, I'm also working with another PhD student, Arnaud Audet, co-supervised with a colleague from the university, Christian Pascaretta. So modeling and prediction of performance in team sports through the study of collective dynamics. Roughly, the idea is that we know pretty well how to model this kind of fish behavior or bird behavior, and we would like to know if the same kind of model can apply to rugby players. Okay, so in Toulouse, probably you know these guys playing in red and black, and I'm just going to show you something. So I hope it will work. Okay, so let's have a look. And it's a try for Toulouse. Congratulations. So now here is the same thing. Okay, it's not so nice to see it on television, but it's the same action, but it has been rebuilt based on GPS device the player has on their back when they play rugby. So in orange you have what is called it's the same team, orange and blue, but just the pack, the 8 tough guys and the others in orange, okay? But you have the same action, okay? You have this race there, pass the ball, and so on, okay? So based on this, we are trying, based on GPS data that we can gather from rugby players, we are trying to see if we can have some kind of modeling of the optimal strategies in rugby. Okay, and to finish, I would like to share other publication I've been involved. So here with colleagues from INRAE, National Research Institute in Agriculture. So we have been working for several years on wheat for to improve the digestibility of wheat. I have worked with a voice therapist to help sport and fitness instructors to prevent voice troubles. These are collaboration with colleagues from INSERM, National French Institute for Research in Medical and Public Health. They are just located near the hospital, near the university, on obesity and what are the consequences on the muscle of obese persons that are subjected to a very strict diet. This was with a surgery that wanted to show how to reconstruct a face that has been damaged, so a symmetric face. So tomographic osteometry of the zygomatic bone applied to traumatology of facial bones and so on. I've worked with colleagues about what could be microbial communities we could put in water to depollute bad water. This was studied with a psychiatrist in Netherlands, and last one with the colleagues, specialists in marine biology, marine biology, to study the development of the brain of a specific shark. Okay, so that's all I wanted to share with you today. So if you have any question, I'll be happy to try to answer them. So thank you for your attention, everyone. Yeah.
So I'm glad that you choose a Montpellier Perpignan site. So do you have questions? Yeah, or from Montpellier or Perpignan.
Could you please remind us of the definition of the Voronoi diagram?
Voronoi diagram. Okay. So here I have just an area with points in here. It would be easier if I could write something. Maybe I can go up there. So, okay. I have this rectangle and I have two nodes, for instance. So I want to build two areas where so everything here is closer from this point to the other one, and everything here is closer. Okay.
J'imagine que oui. Voilà. Est-ce que là vous voyez le tableau si on arrête de partager?
Okay, so the idea.
Il faut que tu dessines dans la partie
ici.
Serrée, ouais.
Okay. Okay, so briefly, we only 2 points in this area. I can, oops. So it's just a way to divide a specific area with here 2 parts. Everything that is like that is closer from this node to this one. Everything there is closer from this one to the other one. And with other points, we can display this kind of
Are there
representation.
specific rules on this? For example, the distances or the mean distance.
Oh, yeah. Here, just I don't know exactly every kind of distance we can use, but here, just considering just mediatrix of this segment and just build this these two areas. Okay.
Are there other questions? So maybe I have one.
Yeah.
So you are working with scientists from various areas, and part of your work is to translate the mathematical results to. And my question is how far you have to understand their problems, and because you cannot go deep into all these problems, I guess. So how far can you go into these problems and to be able to explain corresponding mathematics?
So first, you're right. Big part of my job consists in translating from biology to mathematics, then mathematics to biology. I would say I am not a biologist, but I know I have to understand a minimum vocabulary to be able to translate, like I have tried to sketch. I don't have to be afraid of big words, transcriptomics, proteomics, some kind of stuff, but so I have to be trained a little of biology, I have to be trained a little of psychiatry and so on, but I don't have to become a specialist. And when I organize a training workshop for biologists, I just say, okay, we have 2 days to spend together. You will not become statistician in the same way as I will not become a biologist if I spend 2 days in your lab. But it was what was written at the beginning. I tried to, oops. I have to try to learn the language of biology. As the colleagues in biology has to learn some pieces of language of mathematics. So let's try to communicate at least, even if we do not understand what we do each other in our field. Okay.
Thank you. Are there other questions?
No question, everything was clear, I hope.
Sébastien, tu entends Nadia?
Non.
Alors, Nadia, moi je t'entends, je vais la répéter.
Maybe she can...
Je voulais juste savoir, concernant le Voronoi diagramme, est-ce que c'est possible de l'intégrer au réseau de neurones ou est-ce que c'est déjà fait?
I don't know. I don't know. I'm not a specialist neither in a Voronoi diagram, but I find it just a nice illustration of relatively simple math in a context such as sport and collective dynamics in sport. Okay, actually, I don't know. I don't know if it has already been matched with neural networks. I don't know.
Okay, other questions?
I switch off when you talk, but I switch on if someone else talks.
So,
No?
thank you again, Sébastien.
Okay. Thank you, everyone.